brought to you by dementiaresearcher.nihr.ac.uk in association with Alzheimer's Research UK, Alzheimer's Society, Race Against Dementia and the Alzheimer's Association, bringing you research, news, career tips and support.
Welcome to the Dementia Researcher podcast. I'm Dr Kamar Ameen-Ali, Senior Lecturer in Biomedical Science at Teesside University and regular Dementia Researcher blogger. Today we've got an episode that's going to stimulate your neurons because we're going to be talking about the recent British Neuroscience Association Festival, or BNA for short. And we're going to be bringing you some of the most exciting and thought-provoking highlights from this incredible event. We've got a fantastic lineup of special guests joining us, each with their own unique perspective on the latest trends and breakthroughs in their fields of discovery, from cutting-edge science to inspiring stories of perseverance and discovery. This episode is packed with insights and inspiration for anyone that's interested in the field of neuroscience. So sit back, relax, and get ready to have your mind blown as we reminisce on our week in Brighton. So let's meet our guests. First, we have the incredible Dr Charlie Arber. Next, we have the brilliant Dr Dorothy Tse. Also, the fabulous Dr Dayne Beccano-Kelly. And last but not least, the awesome Dr Nora Bengoa. Welcome, everyone.
Hi.
Hi.
Thanks for having us.
Hello. Hi.
So, why don't you all introduce yourselves properly? So let's start with Charlie.
Okay, hi everyone. My name's Charlie Arber. I'm a Senior Research Fellow at the Institute of Neurology here at UCL. And my work focuses around stem cell models of familial Alzheimer's disease. So trying to understand the early mechanisms of disease with a view to trying to reverse Alzheimer's disease.
Thanks, Charlie. And Dorothy?
Hi, everyone. My name is Dorothy. I'm a senior lecturer at Anchucarro University, also an honorary fellow at University of Edinburgh. I'm a neuroscientist and I'm interested in memory and learning, healthy aging, and Alzheimer's disease. So currently I'm working on to develop a behavioral paradigm in spatial navigation that have a better translation between animals and human studies.
Thanks, Dorothy. And over to Dayne.
Hi, I'm Dayne Beccano-Kelly. I'm a group leader at the UK Dementia Research Institute here at Cardiff University. My work focuses on Parkinson's and looks at trying to mimic the longitudinal timeline of the disorder to really understand how and what changes over time at the synaptic level.
Welcome, Dayne, and over to Nora.
Hi, I'm Nora. I work at ABCN, or Anchucarro Basque Center for Neuroscience, here in the sunny northern Spain. I am a principal investigator and I lead the lab of aggregation and glial response. We're really interested in looking at how proteins like synuclein or tau can aggregate and elicit glial responses in both Parkinson's and Alzheimer's.
Welcome everyone to the podcast. So for those listeners outside the UK or for those who don't work in neuroscience, let me set the scene for you. And please stick with us because I promise you'll still find this show interesting, even if you're not in the world of fundamental science. Now, the BNA International Festival of Neuroscience is a unique, dynamic, and inclusive meeting. As you would expect, it has an incredible scientific programme, but also a real festival feel with fringe and community engagement events across the city, career development talks, workshops, symposia, a silent theatre, rapid fire poster talks, and much, much more. Now, the format for these types of shows is simple. So we're going to go around the studio, the virtual studio we've got today, a few times, sharing our highlights, summarising any talks, posters, any sessions, sharing our takeaways, not literally our food takeaways, but our takeaways from the festival, of course, and generally helping anyone who could attend to feel as if they were there. So I know we were all involved in sessions and events during the meeting, so why don't we start by sharing how these went? So, Charlie, let's start with you. So, if I'm right, you spoke and you co-chaired a session. So, how was that?
Yeah, it was great. I suppose it was the first time that I've been involved in chairing a session at quite a large meeting like this. So I did a little bit of preparation in advance, and we had 2 speakers online and 2 speakers in person. And I suppose the biggest challenge was that we had someone speaking from Uganda, and we either had their voice or their video and never both at the same time. So we had to quickly think on our feet and shuffle the order of speakers. And then thankfully the audio-visual team were brilliant and sorted it for them afterwards. And it was really well worth it, because it was a disease that I knew nothing about, nodding syndrome, which has something to do with tau in African populations. So it was fascinating and well worth sticking with it in the end.
So what was the session on?
It was translational advances. It was a bit of a mix. So I spoke about stem cell models. Someone else spoke about astrocytes. We had nodding syndrome, and then we had antibodies as well. So it was about fundamental advances, and it was great. It was nice to see a mix of topics, all sort of aiming towards the same thing. So it was great. Yeah, I enjoyed it a lot.
I think that's one of the benefits of one of these kind of broader neuroscience meetings, because you do get to mix with scientists that are working on different things compared to what your personal topic might be, and it kind of almost broadens the work that maybe you're going to be involved in, and you can speak with other people that you might not normally speak with because they're not in your direct area of work. Dorothy, I know that you were also involved in speaking and co-chairing a session as well. Am I right?
Yeah, so the session that I chaired is about spatial memory. So it's in behavioral insight into the nature of spatial memory, and it's convened by European Brain and Behavioral Society. So it's linked between like behavioral and also like cognitive process of neuromechanisms. And so there are 4 speakers, like Charlie's sessions. We have 2 speakers online and 2 speakers in person. And so we got 1 speaker from Dartmouth University from USA. So the time zone wise, it's we got to liaise about a bit. And then the symposium itself is about like recent studies in the area of spatial memory. For example, there are recent studies in place cells. And for those of you who don't know what place cells are neurons in the hippocampus that fire when the animal visits specific regions. So we call it like place cells and place fields. And so it's a lot of animal studies, and also one of the speakers talk about humans like studies, cognitive navigation too. So it's very interesting, and I like it because there's like mix of like animal studies and also human studies. So it's a good like multidisciplinary like session, like looking into both like animals and human and in spatial navigation.
I think it's one of those areas of fundamental science where you can see the direct implications. I mean, this is Dementia Researcher podcast, but looking at fundamental science in terms of spatial memory, episodic memory, looking at animal models to determine what those fundamental processes are, can have translation over to trying to understand then in disease models and then in human disease, especially relating to dementia, where we know that some of those early kind of cognitive changes that we see are to do with spatial navigation, to do with episodic memory. So yeah, definitely I can see how a session like that would appeal to people that are both working with animal models and doing human studies as well. Sounds really exciting. Nora, you also were involved in a session as well. Do you want to tell us a little bit about that?
Sure. I was co-chair to the session on techniques and technologies for the study of Parkinson's disease, which was sponsored by Parkinson's UK or PDUK. And I think we had a fantastic mix of different topics there, because we had a talk by Caleb Weber exploring the heterogeneity in iPSC models, which was radically different, for example, to that of Tiago Tedo, who was talking on how we can use perhaps exosomes or ectosomes, which I did not know existed until this very talk, in order to, he presented a biochemical study, but I think the further ahead route of research would be to use these and harness the potential between the different sorts of vesicles in order to use them as biomarkers. And then we had a fantastic talk by Amanda Lewis on the classification of Lewy bodies. And so a bit what Dayne's lab explores, but at the Lewy body level, right? Like if we start with a pale body, which is the very early lesion in Parkinson's, where synuclein seems to start to accumulate, how does that then mature, let's say, into a Lewy body? And she just does beautiful clem studies that just blew me away, to be honest. It's just so nice and such a very fantastic continuation to the early field shocking Shamaradian paper a few years back that it was such a privilege to co-host, really.
And how did you get involved with that, Nora, in terms of the organisation of that session?
Well, I feel like the credit is for Dayne and David Dexter, because I simply laid back and got an email from David Dexter, but I feel like he may have gotten some poking from Dayne. You might have to ask Dayne, actually, because I was just delighted to receive the invitation, and it was a wonderful experience, and the BNA as a whole was a wonderful experience.
Well, shall we naturally hand over to Dayne then?
No, you got it all entirely on your own merit. Don't try and push and deflect this away, Nora. You know you were there on your merit, and that was really good. I was actually at that session. It was a wonderful session, and I can fully concur. All of the talks within that session, including your own, was really good and really refreshing. I did a few things at the BNA. It was quite busy. Full disclosure, I'm co-chair for the BNA 2023 program organizing committee and a joint meeting secretary. So I had a hand in setting everything up, I suppose. Although actually full credit needs to really go to the team, the real people that were doing the work. So like Sophie and Joe and Louise and Danny and Sophie Grange. So they were really the ones that put everything together. But I did end up running around a little bit. I was chairing a couple of the plenary sessions. One for Joanna Wardlaw, who has done some fantastic work across the course of her career. So she was rewarded with the John Wollstonecraft lecture, which celebrates leaders in the field of sort of neuroscience and neurotransmission. And she was talking about her work involving Lackey too. So she was looking at the translation of the work that she's done into clinical trials, looking at how to help with lacuna infarcts and the vasculature within the brain, and how this can then have a knock-on effect to improving cognition, which really spoke to me way back when I did my PhD. I used to work on stroke and the onset of Alzheimer's disease, so I really liked that talk a lot. And I also got to equally have another plenary chair of another plenary session, which was Dimitri Kuhlmann, which is another honor. He's done some great work on electrophysiology, excitability. He's a neurologist, and he was talking about some of the work that he's going on to do to make trials using really fascinating stuff with driving or silencing the activity of neurons based on an activity-dependent manner. So using potassium channels to drive silencing in those neurons, which then actually increase in activity at the time. Excuse me. And then I'm sure we're going to get onto it later, but I was at some of the other satellite areas, so some of the speed dating and the careers talks and sessions, and I thought it was really good. It was really fun. It was great to interact with lots of people. As a lot of people said, it was great to see people in 3D. I know we're doing this online; it's a necessity here, but it's great to sort of meet and chat with people about it in real life. So, and remember how short and tall everybody is, and in real life. So that's always good.
It does make a difference, doesn't it? And I'm quite a short person, and it's the first time that I was meeting some people in real life as well. But I can always assume people are going to be taller than me. So, but I think maybe they get shocked with how I'm like only 5'1".
See, I think I imagine myself like taller than everybody else, and I get the shock of my life when everybody's towering over me, and I prefer it online now. It's better for
I also want to mention, so Dorothy and I were also involved in a session that was co-organised by the BNA and also the Arber Network, which is an organisation that promotes diversity and equity in neuroscience. And it involved us and others kind of running roundtable discussions on topics such as careers, work-life balance, reducing unconscious bias, things like that, and then co-producing solutions with those who attended. Because I think a lot of the time when we talk about these issues that aren't necessarily specific to neuroscience, but more to academia more broadly, we're highlighting what the problems are, but not very forthcoming with how we solve these problems. And actually, if there are any solutions, it tends to come top down. So the idea of actually co-producing solutions, I thought was really innovative and exciting. So Dorothy and I were involved in this session with those who attended. So Dorothy, why don't you tell us how you found that session went for you?
Yeah, I found it's great because it's a workshop too, right? So it's like we divide it into like 8 like tables, like round tables. So we discuss like different topics, and then we share, and then we rotate and then discuss like further of these topics. So as Kim mentioned, like the topics are like about work-life balance and career paths and like job security, which I think is important, like especially for early career researchers, and definition of excellence and mobility and unconscious bias. So there are lots of like diverse topics we discuss. And Kim, I think you hosted at the career paths session. Yes. So with me, I did like on the work-life balance. I found it's hard also. Like I did some homework before I chair of this session. So I think it's important to find out what is the core things that you like to do it in life, and then you make sure you do them first before like you have these other like small tasks occupied you. So yeah, and I think we got like a really good discussion, and everybody just joined in and have some work on some solutions for that. And I remember one of the researchers talking about with work-life balance, right? So we have to like manage our time, prioritize things, right? And we also have some practical solution for that. For example, like zero like inbox, like when you do your emails, right? So make sure you clear it as soon as possible. So just again prioritize. So I think it's good there's some like practical solutions come out.
Yeah, as you said, Dorothy, my table, we were looking at career paths, and so we rotated the tables 3 times. So I had 3 groups, and what really shocked me was the same things came up with each group. So they were sharing that within their institutions, careers outside of academia aren't really promoted within their departments, within their universities. And I was quite surprised by that, because sometimes you see things advertised about things like medical writing or working in industry, but either it's not getting to postdocs and PhD students, it's not advertised as well, or they're just not frequently organised enough. They're kind of sporadic. They're not kind of there enough or promoted enough. I feel some people said that they couldn't talk about careers outside of academia within their group because it was almost seen as failing or not pursuing what maybe their PI wanted. And it's just a shame that people might want to explore but don't feel like they can explore options outside of their PhD. And I think we need to think of PhDs and postdocs as training for a multitude of different career options. And I've written about that a lot for Dementia Researcher. So it was a shame that these same problems were coming up. So it's obviously something that's quite prevalent. But then we also came up with some really, really great solutions. So we know that some funders, as part of their PhDs, they have these almost like internships, I guess you could call them, that are kind of built into the PhD studentships, where they can go and spend some time in a career outside of academia, whether that's spending some time working with a medical writing company or working with industry. so they can actually have some exposure to these different options, not just academia. And I think one thing that came back was actually that should be more widely available to students, but also to postdocs, because I think it is only a select few funders that do offer that at the moment. So we did come up with some good solutions, but it'll be interesting to see now how we move forward in potentially trying to implement some of those solutions. I don't know what any of the rest of the panel think about that.
I think it's definitely a worry that people are not comfortable sharing, that they're wanting to explore things outside of academia. I think from a language perspective as well as a community, we should strive to stop calling them alternative careers. I think it just ingrains that concept that the way is academia and anything else is just the other stuff. And it's not. These are careers. All of them are careers. And with my team, for example, I ask them, not on a daily basis, but every so often. Maybe they think I nag too much, but I do ask them quite regularly, what do you think it is that you want to do later? And I do explore a lot of the different options with them, and I do try to give them, so as PIs, what I mean is we can give them our contacts. I'm sure everyone, or almost everyone, who has gone to industry or to academic writing or to medical writing or to these any type of career option that is not academia, is more than happy to spare some minutes and even just have a conversation, because the placements seem like a fantastic opportunity, but I can see how that is difficult to make widely available to everyone, right? But just to have a conversation over Zoom, these days everyone, will engage in that. And just for us as PIs to promote that as much as possible, and to try to convince early career researchers or ECRs to just really don't be afraid to ask, because if you don't ask, you don't get. But also you really shouldn't be afraid to ask. And if you feel like you're not comfortable with your PI, just, we're all online. I'm sure this panel here is more than happy to help. So just reach out and we'll be happy to help.
Absolutely. I completely agree, Nora. I think that what you said there, you shouldn't be calling them sort of, alternative careers. They are just careers. They're all careers. And another point is when we send off, PhDs and postdocs to another lab, and we see it as growth for a network, we now have ties to another network. The same can be held true for moving into different areas, right? So if I have friends that have moved into the Go Science, so the Government Office for Science in the UK, and that ties to politics and government and policy design is very vital for a lot of the stuff we do, and that's very useful as well. I have friends that have moved into and colleagues that have moved into IP and development of and ideas development, and this is also really useful for if we have patents or if we have other things coming out. So we shouldn't be so narrow-minded as just to think that the ties that we can form only consist of other academic institutions. And I mean, I've put people into contact with these people before, and at PhD level, at postdoc level. Even when we were at Oxford, Nora, I ran a program where we did talks from these external sources so that you could, show that it's not only academia or bust, right? There are other ways and means of doing things. So I'm really interested to know, Kamar and Dorothy, did you, the people that were at your table, was it one particular sort of age profile or demographic for was it like only PhDs, or was it PhDs and postdocs, or were there a few, occasionally, some PIs there as well?
There was predominantly PhD students on my tables and postdocs, but interestingly, we did have a few people that worked outside of academia already. So it was really good to get their perspective on what their experience was like moving outside of academia and kind of how they transitioned. So it was a bit of a mix, but I don't think I had any PIs on my table. I don't know about you, Dorothy.
Yeah, so in my table, so at 3 rounds, there are some PIs, not majority, and there are some students, PhD students, but they have prior experience in working in industry before they go to PhDs, which is very interesting because it gives a different perspective. Because we talk about like long hours, like working long hours in academic. So again, because it's linked to like work-life balance. So one of the key themes we talk about is like long hours do not equal to productive hours. So I think that's like hitting a lot of like people like with the work long hours in the lab, but sometimes taking a break and setting boundaries are also important. And I also would like to echo what Nora and Dayne mentioned. I think it's important also for the PI, to actually encourage or like support, the students to look into different career options. I found, because during this discussion, some of the PhD students mentioned that they have not, like they were worried to like spend time, outside whether they doing experiments, to look into different career options. But yeah, so I think it's important, maybe as a role model, or like support your lab, to grow in that way. Mm-hmm.
Can I just pick up on that? I think it's just amazing that we're talking about the career development, and it was my first time going to BNA, and I wasn't really sure what to expect, but it was one of the massive highlights that there was so much thought and community spirit behind it. So as well as career development, for example, I went to a CV clinic because I never knew 6-page CVs were normal in science, so it's good to know. But as well as that, there's the open access with the BNA's own journal. There's sort of credibility and reproducibility, and there was a whole plenary session on credibility and reproducibility that was amazing. For example, what represents an N number and reproducibility between experiments, which was really interesting to discuss. And and even pre-registration, which I hadn't really considered, but for all topics, pre-registration for even a basic science experiment, what are you expecting? What are you hoping to get out of it? I think was really interesting to hear, because sometimes looking back after an experiment, I did actually do what I said I was going to do, and you might not have the same sort of you might have more positivity after an experiment than you thought. But I think it was just amazing to see the BNA as a society, sort of all these extra things outside the academia, which I was really impressed by, and I loved.
I agree, Charlie. They're definitely pushing forward on a lot of those different agendas, and they're leaders in a lot of those things, definitely. Since we're talking about careers, Dayne, can I come back to you? Because I know that you're involved in the work in the career zone and in the career speed dating as well. Am I right? If so, how did that go for you?
Those were excellent. It was my first time speed dating, and it was an excellent experience. I really enjoyed myself. Now, I met some fantastic PhDs and postdocs at the speed dating session, sat at the table, and obviously the idea is you rotate round and have quick chats with everybody. It was really good to see a breadth of idea sets and questions, and really get to sort of chat with the wide variety of people. That's another bonus of the BNA, I really think, is that you get to talk to, neuroscientists, not just from Parkinson's and not just even more niche from sort of like the synaptic or electrophysiological angle, which sometimes is the realms that I walk in, but to talk to structural protein biologists and just fundamental mechanisms of science. And it was really, really refreshing and interesting to hear the same sorts of questions coming from them as the students and postdocs that I usually see. How do you progress at the career ladder? What would you look for in a postdoc? What would be the next step for me? How do I approach and apply for fellowships and funding, etc.? But it was just, there was an energy. There was a real nice energy about it. Everybody was enjoying themselves. They were eager to talk, eager to ask questions. Nobody felt I feel overawed by talking to anybody. In fact, what I heard back from a few of them at the speed dating was that they couldn't believe there was people at higher up levels, like Patrick Brunding or David Dexter, walking around and just engaging and interacting with them. And I think they were a bit starstruck in some cases, which was great to hear, because that's good that they were having such good interactions. And then sort of the career clinic was similar. It was people coming up and talking about what they were trying to do, what they were trying to achieve. We were next to the CV clinic, Charlie, and there was lots of people there also. And yeah, it was good to see that there was, at least from my perspective, a lot of engagement with what could possibly be considered the sort of more satellite things at the meeting, not just the talks, but these extra bits. And there was a lot of engagement with that, which I think was really, really, really good. It's exactly what we would have wanted.
Sounds fantastic. So what I would like to kind of ask you about now is any other sessions, any other posters, events that were particular highlight for you. So let's start with Charlie. So anything that you kind of experienced during the meeting that was a particular highlight for you, can be a particular session, any particular posters, or any of these kind of satellite events that we've just mentioned.
So I think one of the talks I wanted to see the most was probably the first talk on the first day, Sunday. So I caught the very first train from my station at home and only missed the first 5 minutes of the first talk. But it was around immunotherapy and Alzheimer's disease and other diseases. And James Nichol from Southampton was presenting data on some of the older trials with immunotherapy and Alzheimer's disease, where the drug was AB1792, and what they did was inject amyloid into people, evoke an immune response, and that then clears plaques from the brain. And those trials were 10, 20 years ago now, so some people have passed away since then, and he's looked at the pathology in those brains. And what's interesting is obviously the plaques are cleared away, but also some of the microglial activation is reduced, and the pathology seems like there's been a positive effect of some of these drugs. And the reason this was such a highlight now especially is because of the news at the time of recording just yesterday, but when this goes out last week, around donanemab as the second passive immunotherapy, I suppose. So this is giving the antibodies rather than the antigen. Donanemab is the second antibody, 2.5 I'd say, after aducanumab, but we now have 3 antibodies that can effectively clear away plaques, and 2 that effectively show cognitive slowing of cognitive decline, which I think is really exciting. And it's taken 20, even 30 years since the discovery of amyloid, but we're really on the cusp of something exciting now. And I think looking at some of these pathology and some of the results from the trials 10, 20 years ago is really fascinating about how it works and what we need to do to take this forward. So I think that's really one of the highlights, was to see, the state of the art and where we are with new therapies for Alzheimer's disease.
So a highlight at the very beginning, pretty much, of the meeting. Sounds really interesting. So Nora, what about you? What was a particular highlight that stuck out for you?
I think one of the highlights that I'd like to kind of mention, which has tangentially already been mentioned, is basically just the diversity in the program. So it is very easy to just talk about the neuron and just have neuron-centric talks. And when I talk about diversity, people naturally go to EDI and inclusivity, but I also think about it in the topics that are covered, right? And just to have that so much glial research in the program, to acknowledge that these cells are really important in neuroscience, that we just should not be focusing on a subset of them, and to explore all that is just absolutely fantastic. And there was a PDUK session where Hazel Hall-Roberts and Julia TCW and Emma Mead, amongst others, were really not only looking at the biology of microglia, but also using these cells for screening assays, because we've been focusing on the neuron a lot, and we've just not paid attention to the glia, and to just be using them now in functional studies and to look at drug discovery, I think is fantastic. But I also think it speaks to the organizing and the building the program, right? Because if you don't plan these sessions, we simply miss out on these talks, and we don't get that diversity in the cellular context of the brain.
Yeah, I'd just like to agree wholeheartedly. That was a fantastic session, and hammering your point home, I think Julia TCW's results showing that you only see phenotypes when you co-culture the glia with neurons, and you don't see them when you grow these cells on their own, I think really, supports what you're saying.
Exactly. For example, Parkinson's and Alzheimer's, they're multifactorial diseases. If it was a Mendelian P, if you will, if it was just down to one gene, we would already have cracked it, and we haven't, right? So we just need to build in that diversity into the programs of the scientific meetings, our models, everything.
Yeah, really good point. I think one of the challenges is with a broad neuroscience meeting is having these parallel sessions and covering all these things, because I certainly found that there was several different sessions that were happening at the same time, and found it really difficult to pick the ones that I wanted to go to, because there was several happening at the same time that were really relevant for what I'm interested in. So Dorothy, what about you? What was a particular highlight?
Yeah, I would like to echo with Nora and Charlie. So I think when talk about diversity, apart from like topics, I think it's the elements also with diversity. So I think one of my highlights is the last talk is on Colin Blakemore. I found it's like quite moved. Like not only like talk about like how amazing like Colin's work was, like the importance of like the plasticity in the brain and focusing on postnatal plasticity of visual system, because that's he's amazing in doing that. It's also, you can see people previously worked from his lab actually presenting like a lot about his work, and also he was a great mentor. So I think that elements like moved me a lot. Because Tara Spice-Jones also talked about that. So I think it was great. Yeah.
Thanks, Dorothy. And Dayne, what kind of stuck out as a highlight for you?
So I just complete the set. I'm really pleased to hear that everybody really enjoyed the breadth of the types of talk, the types of sessions that were held, and the different platforms with which that was given. So the posters and the talks and the parallel sessions, and then things like the speed dating. So that's really good to hear. One of the things behind the scenes that we, just to give you a little insight, Kamar, that's exactly what we wanted to hear, is that you couldn't pick because some of the parallel sessions were so good, rather than, well, none of them were that good, so I went for a walk down the beach and went to see, like, getting some fish and chips alongside the pier just for this particular session. It's good that there's so much to see and to do. That's right. So, and I'm glad that it was like that, and I felt like that. At one point I was jumping between parallel sessions, which was hard. Trying to gauge it and write all these notes down, which was really good. But one of the real highlights for me was watching Patrick Brindon talk. Not just because of the science that he was doing. So he was talking about synucleinopathy and synuclein-based models, but he was very balanced about the idea of synuclein and its impact in Parkinson's. That it's of its import, but not that it's solely to do with only synuclein. And sometimes things can become fairly dogmatic in the field, and it's really nice to hear from somebody so eminent as himself that there are other ways and means, and these were more possibly could be modifiers of the disorder, as well as driving forces. There's a spectrum, as there is across all of neurological conditions, and it affects people differently, and we see different outcomes when we look at the pathology, and therefore it can't be, as Nora just put it, that Mendelian P. It can't be just that one thing that's causing the same thing, because we get such breadth of symptoms and histological pathology. So it was really nice to hear somebody like that say it on stage, whilst then talking about his model system, which is heavily based on synuclein, which it just provided a nice perspective. The other great thing was, as well as I say, as well as the data that he was doing and showing, which was really interesting with infections and bladder infections, UTI, maybe being one of the places where things like MSA could be stemming from, etc., was his assertions on two fronts. The first is, we don't want to just drive towards that endpoint histopathology all the time. Sometimes progression is the key, and coming and looking at my particular work and angles, perhaps I'm biased, but trying to look at progression and progressive models is trying to mimic the disorder in general, rather than just to hurry to the end point, is quite nice to hear, because he had a model which needed a lot of time before it developed any sort of histological pathology, and the journals were perhaps a little hasty to reject work or to question his work, which brings me on to my second point, in that he was quite clear with his viewpoints on keeping postdocs waiting for 18-20 months on decision makings of journals, just because somebody wanted to do everything slightly to the right again in a mouse model and correct it, having seen that this already took a long time, and perhaps not trying to look for a paper that explains everything in one foul swoop, and instead maybe perhaps allowing people to break up their data and seeing it for the merit of what is being presented at the time. And I thought that was, brave, and it was also something that people wanted to or needed to hear. So his talk to me spoke on multiple different levels, not just on data, but about how the concept and the culture of science should perhaps be being changed. And so it was really lovely to hear that plenary talk. It was great.
It's always great when you come away from a talk or a session, not just taking away the scientific content, but these are the things that you can then take away and apply in kind of your work life as well. I think for me, one of the sessions that I really enjoyed was the one on neurovascular contributions to dementia. So understanding how blood changes to blood flow in the brain might have a role in the onset of and progression of certain neurodegenerative diseases like Alzheimer's, and how that can then have particular implications for potential treatments. And I found that what really stuck out for me in that session was how we had early career researchers speaking alongside really eminent distinguished professors, and them sharing a platform. I thought that was really nice to see, and also bringing early career researchers on to co-chair these sessions as well and co-organise them. I thought that was really nice to see. So it was really exciting to see Beth Eyre, who's also one of our Dementia Researcher bloggers, and she was talking about her PhD research. But we, in the same session, had Professor Edith Hamill from McGill University. And I just thought it was really nice to kind of see them sharing a platform together and talking about the same topic. And I think it was the first time I'd ever seen anybody get a round of applause at the start of their talk, because Professor Hamill is so distinguished in her area, and she had a round of applause at the start of her talk, which I thought was quite funny. I was like, imagine if I get there in my career where I'm applauded just by existing. That was quite exciting.
Coming on stage like a rock star.
Yeah.
Exactly.
Yeah.
It's what we all strive for,
Exactly.
right?
We can all dream.
But then I think as well, something aside from the scientific content that stuck out for me was, did anybody go to the peer review section? And when I say peer, for anyone that's listening, it was spelled P-I-E-R, because obviously we were at the seaside. So did anybody go to that session? Because I remember walking past the conference the day before it opened and seeing these kind of fairground tents through the window. And I was thinking, what are they doing in there? So did anybody attend that? I want to kind of explain to our listeners what that was. Dorothy, did you go to the peer review?
I did. It was great fun. So first of all, so it's very colorful. I'm just trying to describe, like, it's very colorful. It's like in a circus. I don't know, is it the word right? Yes, yeah. And I found it's amazing. So you did it with Susanna Walker, who's the Liverpool Neuroscience Group lead. So first of all, we need to hook a duck. So, and under the duck there, at the bottom of the duck there is like different topics, there's important topics in the neuroscience field. So let's just say, for example, the topic that I did was how do we actually influence like the policy, like about neuroscience, or let's just say if there's not enough funding, what can we do about it? So there are different topics, important topics, and then we have to do some activities like throw like a ball towards like some coconuts. Yeah.
I think it's called a coconut shy.
Coconut shy, yeah.
Yes, that's probably yeah. So and it's great. It's great fun. And then so we did a lot of like activities. And at the end, I found it's nice is that we got to add some water like to a tank. So it's like every neuroscientist a small step. And it will influence, like, like we make a bigger step. So the idea is that we got to like make the floating brain like out from this big tank. So each of us like contribute a little bit, like small step, and then like together we like have like a big step. What about for those of you who went there? Do you feel this similar things?
I briefly went there and engaged in like you said, you explained it wonderfully. It was just really good to have sort of a bit of fun and stimulate talking points and defunding the NHS, and how the NHS and more interaction with it would be good for neuroscience and neuroscientific research. And I think you did a wonderful job, Dorothy, of describing it. It was a bit of fun, but a way of stimulating some very real topics and real conversations that should be being had by neuroscientists on a regular basis. Just as a not ominous at all, but did you hear that the that giant tank did the very end just and it shattered and all the water came out. So there was a mad dash to try and sort of like sort that out at the very, like literally like just before the last session, I think, and it just shattered and all the water started spilling out. That's nothing to say that we're not to get stronger together and we're not doing so. It's not going to come crashing to a halt, but it was just a bit of it was a bit of fun because it was literally all hands to the pump, but it was interesting.
We can break down barriers together.
Oh, there you go. So there's the spin.
That.
Brilliant.
That was wonderful. Yeah.
Yeah, it's just how we frame it. It just shows the power of neuroscientists when we come together to solve these issues, right?
That. We'll feed that back.
Yeah. Well done.
Good, yes.
Nicely put.
Yeah. It's kind of a good way to kind of end this podcast on, I think. And it was a lot of fun. I've never seen anything like that at any conference that I've been to. And it was a really good way to stimulate discussion because people were just naturally drawn to see what's this big circus tent and what are all these games here for. And it was a lot of fun and a really great way to kind of have those discussions on various topics. So I'm afraid that's all we've got time for today. However, if you want to continue to catch up, or if you're interested in knowing more about the event or watching some of the recorded sessions, head to bna.org.uk. The next event will be in April 2025 and is heading to Liverpool, so we hope to see you there. I would like to thank our incredible guests. So we've got Dr Charlie Arber, Dr Dorothy Tse, Dr Dayne Beccano-Kelly and Dr Nora Bengoa. I'm Dr Kamar Ameen-Ali and you've been listening to the Dementia Researcher podcast. Bye everyone.
Bye. Thanks.
Bye.
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 Brought to you by dementiaresearcher.nihr.ac.uk in association with Alzheimer's Research UK, Alzheimer's Society, Race Against Dementia and the Alzheimer's Association, bringing you research, news, career tips and support.